And on the third day he rose again. Happy Easter. So in a real-life story of resurrection, scientists have recently just restored cellular activity to pig brains hours after they have already been dead. They did this by taking the brains of the pigs and placing them inside of an artificial fluid after they were killed at a slaughterhouse. Specifically, they used a hemoglobin-based acellular, non-coagulative, ecogenic cytoprotective perfusate that promotes the recovery from anoxia. Now, this is a huge breakthrough in the field of science because this is challenging to the long-standing idea that loss of brain functionality in mammalian systems is something that is irreversible. You're dead, you died, you're done. But something important to note is that there were no signs of widespread neuronal activity. Essentially, the level of nerve activity that would be required for there to be some level of consciousness in the animal. But the individual nerve cells were still carrying out metabolic processes, taking in things like sugar and oxygen and spitting things out like carbon dioxide and firing action potentials. And so from a neuroscience perspective, this is a really interesting study into that kind of gray zone between living and dead animals, humans included. Any of you who are familiar with someone who is in a coma, we have the technology today where we can keep a human's biological system still pumping blood to all of the organs and keeping organs alive, even in the absence of neural brain activity that would suggest there's a consciousness to the human. And it's an interesting dilemma because you have people that say, well, is that person alive or are they not? It's also probably worth noting that the pigs that were used in this study were from a slaughterhouse for commercial farming. In other words, these were animals that were being killed for our food supply and livestock, and the brains were donated to the scientists that worked on this research. These pigs were not laboratory pigs that were raised and killed for this purpose specifically. So basically, the scientists took these pig brains that they got from the processing plant, and they put 32 of them in this artificial brain fluid that allowed blood to flow through the blood vessels of the brain and carry the needed nutrients to all of the cells. And some of the areas of the brain that showed healthy neurons that were firing action potentials that was super exciting included the hippocampus and the prefrontal cortex. These are exciting because the prefrontal cortex is kind of like your higher order thinking brain that's involved in complex thought, and the hippocampus is thought to be heavily involved in the consolidation and formation of memories. Now, this artificial nutrient-carrying fluid that the brains were placed in also contained a compound that was designed to block neural activity. And this was done specifically to limit the amount of potential neural activity that could happen in the brains, because the scientists were afraid that too much could be harmful to the brains. So what's really interesting now is that scientists are curious if the neural activity would have been more widespread in the absence of that blocking compound. And so what is the end goal of this research? Are people trying to create a real Frankenstein world where we are resurrecting our dead relatives? Well, no. But if you know anybody who's ever suffered through a stroke where there is a lack of blood reaching the brain, you might know that certain regions of brain tissue can start to die off very, very fast. And the long-standing hypothesis has been that once you lose that brain functionality, it's kind of lost. You cannot restore those cells. What we have known about in neuroscience is that the brain has a level of plasticity to it, and sometimes other regions of the brain can pick up new functions to take over for those lost sections. But in the wake of this study, where we've shown the ability to restore cellular function to essentially thought to be dead cells hours after they have been deceased, this acts as a huge breakthrough for potential future ways to treat brain damage that patients may have suffered in a stroke. And that's kind of cool. So there's a cool little story about pigs and brains that you can share over your dinner tonight, where you cut the ham with your family, if that is your thing. As always, thank you guys so much for watching, and stay inquisitive. Happy Easter.